Okay, welcome to this video of Unit 3, Area of Study 1, Biology. This video is focusing, as you can see, on membranes and transport. This is part of the study design, of course, and if we back out here, we can have a quick look at where it sits on the study design. So this is outcome 1 of Unit 3 on the study design, and we're skipping ahead to this dot point here. We're going to come back to the first dot point in a little bit, but right now we're looking at the structure and function of the plasma membrane and the movement of substances across it. There's a few other sub-points along here that we'll get to along the road as well. Hopefully this should sound familiar because it's certainly something we've covered quite a lot already in Year 11. So it shouldn't be too much new ground in this topic. Okay, so let's get into it. We want to know about the function of the membrane. And there really are many functions of the cell membrane, or as we can see here, also known as the plasma membrane. Biology loves having multiple words for the same thing. Cell membrane, plasma membrane, the same thing. We know that they form the boundary around all cells. It's a feature of all cells. Animal cells, it's the outermost boundary. Plant cells, not the outermost. We've got a cell wall around there as well. So anyway, we know that there's multiple functions of this plasma membrane. Heaps and heaps of different things we could talk about. But there's really only a need to point out these kind of two key features. If you're asked to describe what the function of the plasma membrane is, it is that it selectively controls the movement of substances or materials into and out of the cell. We could also say that it's responsible for cell-to-cell communication or recognition, and we'll look at that certainly in more detail when we look at our part of the course on immunology. The cells have lots of important markers on them that allow them to recognize things in their environment. For now, though, we're probably looking at the movement of substances in and out of the cell. That's the key thing that we're focusing on here. Now, hopefully we'll know that the reason it can do that is because the cell membrane is a semi-permeable membrane, meaning that some things can cross it, some things can't. And for certain substances, they'll have specialized ways, like those protein channels, of getting in and out of the cell. And the cell is able to therefore control the movement of different substances. In order to understand that function, you obviously then need to understand the structure of the membrane. That's a key thing. Now, the membrane it's tiny, it's a very small thing. We can't actually see it. So we use a model to help us describe how we think it's structured and how we think it operates. And at the moment, our best understanding of that is the fluid mosaic model. And the study design says this is what we need to know about. We need to understand this model. So it shouldn't be anything new. The fluid mosaic model tells us that the membrane is largely made up of a phospholipid bilayer. So that is these two layers of phospholipids facing opposite to each other, making the boundary of the cell. And on one side will be the extracellular environment, that is the environment outside of the cell, and the other side, the intracellular environment. These phospholipids are arranged with their hydrophilic tails facing inwards, so squiggly little tails all face inwards of the two opposing layers, and their hydrophilic tails, the things that don't mind water, they're water loving, they face outwards because those outer environments, so the environment outside the cell and the environment inside the cell, are watery, and so it makes for a nice happy medium for this structure, allows us to be very stable and sound. And we'd kind of say that is the fluid component of the membrane, because those phospholipids are not bonded together. You know, you've heard of like covalent and ionic bonds and things like that. Well, there's no covalent or ionic bonds connecting these different molecules. They're just, we say, aggregate together. They hang out together because they've got similar properties, similar likes and dislikes. Mainly we're talking about water there. So they aggregate together and therefore they move quite a bit. If we're able to look at the plasma membrane, if we're able to see it, it would be moving up and down quite a lot. It wouldn't be very stable, it would be fluid, it would be constantly moving. The mosaic part comes about because it's not just phospholipid bilayer. There are other things dotted throughout to create this overall picture. Mosaic, some kind of art term, I don't really know much about art, but I think it means lots of different things coming together to form the one image. Don't quote me on that. But the different things in here are things like cholesterol and in particular the proteins. There's proteins just sticking in the side of this membrane that might be there for cell-to-cell recognition, but importantly there's also these what we would call protein channels, or sometimes we might see them referred to as transmembrane proteins, or sometimes we might see them referred to as ion channels or protein pumps. Lots of different names, and there are specialised types of them, but their role is, as we should know, they have a specific shape and they allow specific molecules to pass through into and out of the cell, part of that selective control of movement. And so that is the mosaic part of this, this fluid mosaic model. See down there, the last line we've got here is that there are a number of other things that are important to membrane structure too. Let's look at some of those. Some of them not so important. I mean, we already know about the protein channels down here. At the top, we've got glycoproteins and glycolipids. Similar sounding names because they've got a similar structure. You can see that each of them have a number of these hexagonal chains stuck on the end of them. Whenever you see a hexagon like that, it's glucose. So each of these have glucose chains stuck on the end of them. And that's where the glyco part of the name comes from. Glyco often refers to glucose. So the glycoprotein here is a protein molecule that's stuck into the membrane with a big long chain of glucose stuck onto the end of it, a glycoprotein. This will have lots of different functions, but we'll look at these in more detail when we look at antigens, which are part of the immune system. Glycoproteins can often be antigens of the cell. Glycolipids we probably won't look at much at all, but hopefully from the name there you can see that it is a lipid. There's a squiggly little line down the bottom there sticking into the phospholipid bilayer. It's a lipid with glycose stuck on the end or glucose chain stuck on the end. Not too worried about those. Cholesterol, it's worth knowing about, and cholesterol can seem a bit confusing sometimes because if you read up on it, it might tell us that cholesterol helps to make sure that the membrane doesn't become too rigid. But then you might also see that it says it helps us to make sure that the membrane doesn't become too loose and doesn't fall apart. And I guess that we're looking for a happy medium there, somewhere in the middle. And cholesterol, we would say, allows the membrane to be nice and fluid. It improves the fluidity of the membrane or keeps it nice and fluid. That's about all we would need to know. You don't need to know the how and why of that, just that it helps it be fluid. Okay, so speaking of membranes, the cell membrane is not the only membrane we'd find in the cell. We know that eukaryotes are made up of lots and lots of membrane-bound organelles. So we've looked at some of these already as listed down here. All these organelles are made up of the same type of membrane. It's always a phospholipid bilayer. And that's really important because it allows these different organelles to exchange vesicles from time to time. So as the endoplasmic reticulum buds off a little vesicle so that it packages up an item, some proteins in a vesicle, and sends it over to the Golgi. Let me get some pen action happening here. So we get some materials, there we go, packaged up here in the endoplasmic reticulum, sent over to the Golgi in a transport vesicle. That transport vesicle will then fuse with the Golgi. And that's fine because it's made up of the same membrane material as the Golgi. They're both phospholipid bilayers, so they can fuse together or bud off different parts, and it's no big deal because it's made of the same stuff. Just as that Golgi can then form a vesicle with some proteins inside it, and it can send that off to the cell membrane to release its contents as that vesicle fuses with the cell membrane. And again, that's no problem because they're both phospholipid bilayers, and so they can merge together, and no concerns there. So keep that in mind. We're dealing with the same membranes all throughout the cell. Now we know a bit about the structure of the membrane, we want to know about the different ways that substances can get in and out of the cell. It's really important that the cell can take in substances so that it can make new molecules, it can make new biomacromolecules, but it's also important that it can then export them out of the cell, or that it can take up ions and water and release them as need be as well. And because the cell membrane is a semi-permeable membrane, there needs to be a number of specific ways of getting these substances in and out. There's these 5 key ways that we need to know about. Again, we've looked at these before, but just to recap, we can classify these as being passive or active modes. Passive modes don't require any energy, so that includes diffusion, facilitated diffusion, and osmosis. Those modes that are active require an input of energy, and that's going to be in the form of ATP, which is our great energy molecule that we find in living organisms. Those include active transport and cytosis, which you'll sometimes see just written as bulk transport. Okay, let's have a look at some of these and recap what we already know about them. Diffusion is a general term. It's not just related to cells. Diffusion happens all around us. It's a natural process whereby molecules will move from areas of high concentration to areas of low concentration. It's the reason that if you release a gas on one side of the room, it will then diffuse or spread throughout the entire room because the particles naturally move from areas of high concentration to low concentration. Doesn't require any energy, just naturally happens. And so substances can move across the cell membrane. They can naturally diffuse across. Not all substances can, and in class we'll look at some of the rules a bit, once again, we'll look at some of the rules in a bit more detail about what governs who can and can't diffuse across a membrane. But small things like oxygen gas or carbon dioxide are the key ones, can easily diffuse in and out of the cell via following their concentration gradient. And that's probably what's happening on the right here. The blue circles here might be oxygen. It's a high concentration on the left-hand side, and so they're diffusing across the membrane to the right-hand side. And then the opposite here, maybe this is carbon dioxide. There's a high concentration on the right-hand side, and they're diffusing to the left. There is a net movement. Now, once the kind of concentration on either side equals out, so once the concentration of blue circles, oxygen, on both sides of the membranes equals out, then there will be no net movement. Now, particles might still move from one side to the other, but the rate at which they're moving from one side to the other should be equal, and therefore the concentrations will stay the same on both sides. And this is why it's really, this is how we kind of rely on these principles when we're circulating oxygen and carbon dioxide around our body, and why it's really important for our body to maintain the concentration gradient by using things like a bloodstream to take the carbon dioxide away, so we've always got that concentration gradient present. Speaking of, there's the human lung and bloodstream, just exactly following on. It's almost like I knew this slide was coming somehow, amazingly. So there are a number of factors that govern how much or the rate of diffusion, things like that are written here, don't want to focus on these too much. Concentration gradient, the distance, obviously the shorter, the easier or the faster it is for things to diffuse. Surface area, we love that concept, high surface area to volume ratio increases the amount of diffusion. And physical barriers, the thinner the barrier, the easier it is for things to diffuse across said barrier. There's a funny picture of phospholipid bilayer. I think we'll look at that one in class probably. Okay, very similar to diffusion in name and in principle is facilitated diffusion. It operates on the exact same concept that molecules move from an area of high concentration to an area of low concentration, but in this case facilitated diffusion refers to it occurring through a protein channel. And that's one of those transmembrane proteins in the cell membrane. These protein channels are selective in what they allow to pass in and out of the cell though. And they do that by having a specific shape, a specific structure that will only allow certain molecules to pass in and out. The cell doesn't want everything getting in and out of these channels, only certain things. And the cell will often use these channels for glucose and amino acids. Again, this doesn't require energy because molecules move with their concentration gradients. It does require these protein channels though. Now what you'll notice down here, hopefully my head's not in the way too much, is that we've got these red particles are moving through the protein channels, but there's also one down here that's able to just diffuse across the cell membrane. And this is not that unusual, that sometimes molecules that could diffuse through the phospholipid bilayer also have protein channels for them to use as well. And the cell will make use of this in a number of ways. And we'll probably look at that in a bit more detail in class. It'd be a good thing to look at. That's probably a good spot to end this now because I've only got 15 minutes on my freebie software. So I'll stop it there and then we can come back for part 2 to look at the other 3 modes of transport.